Let's see. Hi there, students. So we are going to talk about chapter 3. Let's see. Oh, if we can. Alrighty, so just a reminder, you guys do have your, excuse me, your first exam coming up next week on Thursday. So as we approach a week until you have an exam, I definitely want to point out that you don't want to cram. You should have a look at the effective learning videos that I've posted on Canvas under course information. Use that info to your advantage. So make sure that if you haven't started studying yet, you really are starting to study. You have a lot of resources at your disposal, like the chapter guides that I've made for you for Chapter 1, Chapter 2, Chapter 3. Learning curve, again, you can go back in and you can do more questions for each chapter. You can also look at the personalized study suggestions based on your performance on different types of questions from the first go round. Remember, in learning curve, one of the things that I love about it is that it will direct you to specific parts of the book that pertain to a question that you might be having trouble with. Chapter 2 and 3, Chapter 1 doesn't have these, but there are these checkpoints questions. They also are really helpful if you can try answering them. Make sure that you're not just memorizing information, but that you can use what you've learned and apply it to new situations, for example. Make sure you can describe processes, explain how things work. Highly recommend using study groups or partners. Absolutely, if you are in athletics and you have study table, plan a time to get together and study with other people from the class. It's just a really good idea. Make sure that you're talking to me about anything you don't understand completely. I know that I've been away a bit. However, I do really want to encourage you guys to reach out via email. I'm happy to even Skype or FaceTime if that makes things easier for you. So please reach out. Let me know what you guys need. I will have office hours on this coming Tuesday from right after class until, well, really until the beginning of my lab at 1:15. So please come by on Tuesday if you want to go over anything at all or just talk about life or whatever. The Center for Academic Success does have tutoring available for this class. I highly recommend accessing that because, you know, it's there for you whether you use it or not. So highly recommend using that resource. Also, I will be posting an exam review Kahoot, and there's a practice version that you'll have where you can practice the Kahoot. It's going to help you make sure that you can answer those sort of questions that will be on the exam. The exam will be a little more in depth. So it's great to practice, and also there will be a Kahoot challenge which will allow you to receive extra credit on your exam. So more about that on Tuesday. All right, so without further ado, chapter 3. So this chapter is all about our other types of macromolecules, nucleic acids, proteins, and enzymes, which are actually proteins. All right. So learning objectives for this chapter, we're going to detail the structure of nucleic acids, apply our knowledge of transcription and translation, which you guys will be getting, understand how the properties of amino acids affect the three-dimensional structure of the proteins they make up, explain the functions of enzymes, and then you're going to be able to actually apply what you've learned about enzyme regulation to a case study I've developed about drug discovery in the fight against cancer. All right, first, nucleic acids. This is probably review for many of you guys, and that's fine. So, but just in case it's not, Your nucleic acids, that includes your DNA and your RNA. These are informational macromolecules. So your nucleic acids have 3 parts in terms of the monomers. So the basic monomer, and I'll go over here, the basic monomer of a nucleic acid is a nucleotide, and it is made of a base. And these bases, it's just shown as an orange box here, but if you go on over, the base can be either a pyrimidine with sort of this 1 ring structure, or a purine with a 2-ring structure. We'll talk much more about these later, but for right now, just remember that the pyrimidines have 2 rings, or sorry, I'm so sorry, the pyrimidines have 1 ring, and the purines have 2 rings. And you have that base, then you have either a ribose or a deoxyribose sugar. Guess what? That's one of the differences between RNA and DNA. DNA is deoxyribonucleic acid, so it has a deoxyribose, and RNA is ribonucleic acid, so it has a ribose sugar. And then attached to, and it is a sugar, so this is, it should look familiar. It looks just like the sugars that make up carbohydrates. So the base is attached to that sugar, and then on the other side of that sugar, you've got a phosphate group. All right, so altogether we've got a phosphate, a sugar, and a base. So a nucleotide is going to be the monomer of nucleic acids. And of course, what do we do with monomers? We put them together to form polymers. Okay, so just like the reactions that we talked about in the last chapter, these are condensation reactions. So when we want to add a new nucleotide onto a string of nucleotides, a nucleic acid, then it's going to be the phosphate group here that's attached to what we call the 5' numbered carbon. That's going to connect to the 3' carbon on this sugar over here. And oh, look what gets released. An H and an OH, which makes water. So it's a condensation reaction just like you've seen before. So again, the sugars are numbered, so you can see the numbers down here. So number 1, 2, 3, 4, and then the fifth carbon is kind of like off on its own little special branch, and then you always have an oxygen right here. All right, so also important, your new nucleotide always gets added so that the 5' phosphate is added to a free 3' end of a polymer. And nucleic acids always grow in this direction. So we can say they grow 5' to 3'. I always like to think 5' phosphate. So if you've got the phosphate end sticking up, that's the 5' end because that's the carbon that's sticking up here. Okay, so 5', 3'. And the name for this type of bond that occurs between nucleotides is a phosphodiester bond. Phosphodiester bond. It's one of those big fancy words that makes you feel like a fancy pants scientist when you say it. All right, base pairing in DNA and RNA. So, interesting thing, purines always bind to a matching pyrimidine, and they're very specific. So, purines, again, are the ones that have 2 ring structures. So, adenine and guanine are purines, and thymine and cytosine are our pyrimidines with 1 ring structure. Thymine always pairs with adenine. And also, interestingly, the number of hydrogen bonds, and these are the rungs of essentially that sort of twisted DNA ladder classic thingy, the rungs are stuck together by hydrogen bonds, just like you've seen between water molecules. Hydrogen bonding, crazy, crazy important in biology, guys. All right, so adenine and thymine are joined by hydrogen bonds, and cytosine and guanine are joined by hydrogen bonds. Now look at these bonds. What do you notice about a difference between an AT pair and a CG pair? Exactly. The AT pair has 2 hydrogen bonds, boop, boop, and the CG pair has 3 hydrogen bonds. Always, 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 always. A is always with T, and C is always with G. DNA exists as a double strand, of course, inside of the nuclei of your cells. And the double strands, so this strand and this strand, are connected by these hydrogen bonds between the bases. And then, of course, they twist around to make the double helix. And again, you can see where the phosphate group sticking out on the end, that's the 5' end, and then the 3' end is up this way, where there's the 3' carbon with nothing attached to it other than the OH here. And then on the other side, you can see that the phosphate group's at the top here, so this is the 5' end, and down here at the bottom is the 3' end. So, our DNA double strand is what we call anti-parallel. That means that we've got 5' to 3' going up on the left side, and we've got 5' to 3' going down on the right side. Okay, so left and right doesn't really matter, but they are always going in opposite directions, and this is going to get really important in later chapters when we talk more about transcription and translation. All right, RNA, so the other nucleic acids. These guys are just as important as DNA because they take the information from the nucleus. Your DNA never leaves your nucleus. The RNA takes the information from DNA. It's kind of like a little copy machine situation. So these are the copies, and they go out of pores in your nuclear membrane into the cytosol, okay, where they can be read by ribosomes and translated into proteins. So more about that later. Getting ahead of myself. So RNA always exists as a single strand. It is not double-stranded. However, as you see here, this looks much more complicated than that. And that is because RNA can kind of double back on itself and form some complicated structures. All right, so we have accomplished our first objective. Now we're going to work on transcription and translation. All right, so as we already mentioned, your DNA basically has the information for how to build you. And it's expressed through RNA going out of the nucleus and getting read or translated into a polypeptide sequence, which makes a protein. So, DNA also can replicate. When do you need to replicate your DNA? Well, anytime your cells are going to divide, you have to replicate your DNA. You also have to replicate your DNA when you're making your gametes, eggs and sperm. All right, the other thing that DNA can do other than replicate is it can be transcribed through the process of transcription into the RNA, which can leave the nucleus. And then the RNA gets essentially read into a polypeptide sequence through the process of translation. So DNA is kind of our master blueprint, and RNA is the working copy, so the copy that we trust to take out in the world while our real important documents live in a special filing cabinet in our office, for example. Okay, that filing cabinet is the nucleus. So that RNA copy that we take out into the world is what gets read and built and, has the instructions for making proteins. All right, replication. So when you have replication, you're going to start with a double-stranded DNA molecule up here at the top. And when replication takes place, we essentially divide that thing in half and then use those base pairing rules, A and T always bind and C and G always bind, to rebuild a second half. Okay? And we do this on each of the original strands, so then we end up with 2 genetically identical, unless there's a big mistake, two genetically identical double-stranded DNA molecules, and one of each of those can go into each of the new cells when you're replicating, when you're dividing your cells. In transcription, however, the DNA never gets replicated. Some people make the mistake of trying to replicate the DNA before they do transcription. In transcription, we're going to take that DNA code and transcribe it into RNA, which is going to, again, leave the nucleus. All right, so before we go on, let's try this out. So, using your base pairing rules and the fact that the strands of double-stranded DNA are anti-parallel, take this sequence here and replicate it, okay? So if we've divided our original DNA in half, and now we've got a single strand, how do we add more bases using base pairing rules for replication? Okay, so pause this video, do that, label the 3' and 5' ends. Also, I want you guys to try out transcribing it. Now, another difference between DNA and RNA is that the RNA does not have thymine, does not have T's. So RNA has U's instead of T's. Otherwise, it's going to be pretty much the same as when you replicate DNA. Okay? So pause this and try that. Seriously, pause it and try it. All right. So if you actually paused it, what you should get is that if you do replication, you are going to end up with a strand on the other side that is AGT, CGT. So this is just using base pairing rules. So we had a T here, so we know we're going to have an A here. We had a C here, so we know we're going to have a G, A, so we're going to have a T, and so on and so forth, just using the fact that A and T always bind and C and G always bind. And now the ends here, so the 3' end is on this side, and so on our new strand, we know that it's going to be 5' on this side and 3' on this side. Make sense? All right, for real, guys, if it does not make sense, practice, and come talk to me. Shoot me an email, FaceTime with me, whatever you need to do. Transcription. Again, it's going to be a little bit different because we've got U's instead of T's in our RNA. So, again, ignore this replicated strand. But what you've got here in your original is 3' end, TCA, GCA, 5' end. And so when we transcribe this, instead of AGT, when we transcribe, we're going to get AGU because there's no thymine in our RNA, okay? So this is what you're going to get binded with this guy if you're doing replication. And if you're doing transcription, you're going to get this guy, and it's only going to temporarily be bounded with that DNA because it's going to go and leave the cell nucleus. All right, so now we have applied our knowledge of transcription and translation. So now that you know that that works, make sure that you can very, very easily tell me what a new strand of DNA or RNA is going to look like. All right, now we are going to work on understanding how the properties of amino acids affect the 3D structure of the proteins that they make up. All right, guys, I am such a nerd that I have a favorite protein, and this is it. And this is for real what it does. It literally is a little protein. So that orange guy is the protein. Its name is kinesin. And it literally, look how cute it is. It has little freaking feet. And it literally walks along your microtubules, which are sort of the skeleton of your individual cells. and it walks along there, and like a little frickin' atlas, it carries larger molecules in vesicles. So this is a giant vesicle that is full of who knows what, that is probably destined for outside of the cell. So this little Hercules here, he carries that thing all the way from the inside of the cell to the surface of the cell. I mean, look at that thing. It's ridiculous. Kinesin always goes in that direction, by the way. So how fascinating is that? I just love that guy. All right, proteins. What are they? Well, they are polymers. That's kind of the theme here. So this is our fourth type of macromolecule we're going to talk about. But proteins are polymers, and they have incredibly important structural and metabolic roles. They are what I like to think of as the doer molecules in your body. So they do literally pretty much all the stuff, such as facilitate cellular reactions. Those are the special proteins we call enzymes. They provide defense against foreign substances and cells, like antibodies. Did you know your antibodies are proteins? They're signaling molecules. They're signal receivers. We'll talk much more about that later. They're also a storage reservoir, obviously, of random amino acids put together so that you can use them to build other proteins to do stuff. Some proteins, this is getting exhausting, right? They do so many things. So proteins can also provide a lot of structure. So collagen is a protein. Collagen makes up a lot of your tissues and gives them structure, like your skin has lots of collagen. You'll be talking about different types of tissues later in the semester in lab. Oh, there's more. They also function as transport molecules. Hemoglobin is a protein, actually an incredibly fascinating one with quaternary structure, where we stick 4 proteins together to make the full thing. Genetic regulation. So they can literally attach onto your DNA and influence whether or not that DNA gets transcribed into RNA and translated into more of some kind of protein. All right, and the function of these proteins, what all it does in whatever function it has, depends on its structure. And there's a lot of components to the structure of proteins. So the monomers of proteins are amino acids. And every amino acid kind of looks like the diagram on the right there. So they all have a central carbon. Sometimes that's called the alpha carbon, if you ever hear that. They've got a central carbon. They've got what we call a carboxyl group. This sometimes it's written as COOH, sometimes it's written as COO minus, like you have here. It functions as an acid. And then on this side, you have the amino group, or NH3. Funny thing, when stuff's on the left side, a lot of times they put the H's first, so that looks kind of funny. Anyway, so this part has a positive charge, which means it functions as a base. And then here's where the interesting stuff comes into play. The identity of every one of those 20 amino acids is in the side chain. So this group that just says R here, that can have a lot of different chemical structures that we're going to look at. 20 different ones, 20 different monomers, 20 different amino acids make up all the insanely complicated and varied proteins in our body that do all that stuff we talked about. Okay, so the 20 amino acids. So there's a worksheet that I would like you guys to work on. It is posted on Canvas on the amino acids, so please do work on that. You can get together with somebody and work on it. And bring it with you to class. Please bring that worksheet with you to class on next Tuesday. On this worksheet, you're going to be thinking, okay, you're going to be thinking about whether an amino acid is hydrophilic or hydrophobic. How do you tell? Got to think back to what you guys know about polarity. And why does this matter? to an amino acid, and more specifically to a string of amino acids that you might make to make a protein. Okay, I'm going to leave you guys to do that worksheet and think about it. So please pause and work on that worksheet. All right, here we go. The 20 amino acids. This is all of them. Pretty incredible again that we've just got 20 different ones. All right, so one thing that you will notice is that, and again, we're just looking at the R group, so the top part there, this is the same for all of them. And then the R group, the purple thing here, is what differs. So some of our amino acids have a little positive charge. Okay, see that? A lot of times this is associated with an NH3 or an NH that's on the side chain. Okay, so these guys have a positive charge, arginine, histidine, and lysine. And there are two also that have a negative charge in their R group, aspartic acid and glutamic acid. Okay, so both of these are charged, and what do you know about how water interacts with charged stuff? Well, if you remember back to when we talked about salts dissolving in water and what happens to the ions, like the sodium and chloride, you guys know that water molecules will literally surround something that has a charge to it. Okay, so if it's got a positive charge, then those oxygens that are highly electronegative will be attracted to the positive charged parts of these amino acids. And the positive, partially positive hydrogen charge on water molecules is going to be attracted to the negative parts of our two negative amino acids. Okay, so these are going to be hydrophilic. They're very attracted to water. And then if you look in that purple square there, so these guys here, do we see any charges? No. But check out what we do see. All of these R groups have a big old oxygen. What do we know about oxygen? It is greedy for the electron. So usually wherever you see an oxygen, it's going to be highly electronegative, which is going to give you a little bit of polarity. So these are our polar amino acids, and they are also hydrophilic. Now then, we're going to skip these for a second. At the bottom here, we have our nonpolar amino acids. How do we know they're nonpolar? Well, look, they have no charges, they have no oxygens in these R groups, and so they're not polar. So these are usually going to be hydrophobic. And then we've got 3 amino acids that are special cases here. I'll go on to their individual little pages. Okay, so positively charged guys, hydrophilic. By the way, oh my gosh, you guys, you do not have to memorize the 20 amino acids and all their names and which ones are hydrophilic and hydrophobic and blah, blah, blah. I do, however, want you to be able to look at, a chemical diagram and names of some amino acids. You should be able to look at that thing and easily tell me, like, oh, that's a hydrophilic one, oh, that's a hydrophobic one, because blah, 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 blah. How do you know? And you should be able to think through what that means for protein structure. We're going to get to here in a little bit. All right, so there's our charged guys. There's our polar hydrophilic ones with oxygens involved. Yep. Here are our nonpolar ones. No oxygens or charges. Simple as that. All right, here's our special cases, cysteine, glycine, and proline. So if you look at these long enough on the big table, you should notice what is special about each of these guys. Okay. First, glycine, our guy in the middle. Look, it's R group's only a hydrogen. This is a very small amino acid. This is as basic as it gets. It is nonpolar. It fits into tiny little spaces. So Very useful little guy. Proline, the one on the end here. So look at this situation. So you've got your R group, and normally it would kind of like hang down like a little tail, but this R group is sort of reattached to the amino group of this amino acid. So it's got this like ring structure, and that is important because it prevents the rotation of these molecules around these bonds. It actually reduces the reactivity of these molecules. And it's also nonpolar. All right, we're going to stop here for a minute